Hello, everyone. Thank you, Ryan. And as he mentioned, my name is Paul Diehl. I'm the Chief Operating Officer for Selecta. And I'd like to thank everyone for taking time out of their day to tune into our webinar, where I'm going to discuss today about screens using pooled CRISPR and cell barcoding libraries to identify genes responsible for disease biology and moderating therapeutic responses. Before I get into that, though, I'd like to provide a little bit of background about Selecta. Selecta was founded about 14 years ago. We're a relatively small operation south of San Francisco by about a half an hour in Mountain View, California. And the company is very technology focused. We're a private group, and the seed money really to start the company came from small business innovation research grants. And we've developed that into a range of products and services. I'll give you a little bit of an overview of those shortly. Selecta's focus is to enable researchers to identify the genes or genetic elements of interest in various cell or animal models that are driving the phenotypes or biological responses or responses to treatments in these systems, especially ones that are therapeutically relevant or important for disease progression. And in particular, the company is focused on developing approaches to do this that don't require large-scale automation, LIMS systems, other industrial infrastructure. We want technologies that are able to be run in ordinary molecular biology or cell biology laboratories. One of the recent technologies over the last several years that has become accessible that enables this is next generation sequencing. And so most of the readout for the assays and technology that we develop uses next generation sequencing to sort out the various responses that you're looking at and be able to identify and sort through the data and understand what are the ones that are key for the model or system that is being investigated. And we've had a fair amount of success with this approach. There are over 350 some citations on our website. These are publications that we've found that from groups that have either collaborated with us or used our products or services. And it's a searchable database if you'd like to go take a look. Selecta offers a range of products and services based on several technology platforms. Of course, for genetic screening, we have the CRISPR and RNAi libraries, the sgRNA and shRNA libraries, as well as barcoding technology. And we'll talk a lot about that during the presentation today. In addition, we have the driver map targeted RNA expression profiling system. And this is a multiplex PCR-based system to target and amplify transcripts for all of the genes in the human genome, all the protein coding genes, approximately 19,000 protein coding genes. And the products from this amplification then are sequenced on NGS, and it's an alternative really to using RNA sequencing to do the analysis of what genes are expressed in different samples. And since it's PCR based, you can use very small amounts of material down to single cell. The analysis is straightforward because it's really a question of aligning the amplified sequences from the NGS data with the results, and so that can be done in a spreadsheet. Related to that, we have services for digital spatial profiling using NanoString's geomics system to look at expression in different tissue regions or structures on slides. And we have a lot of experience working with lentivirus and lentiviral packaging and viral packaging in general. And so using that experience, we've have a range of products that are pseudotyped lentivirus that have been pseudotyped with the coronavirus spike protein. So this enables laboratories who want to look at spike protein binding to target cells to do so in normal BSL-2 laboratories without having to have heightened biosafety levels that you would need to have if you're working with actual coronaviruses themselves. And then we have custom services based around viral packaging, viral vector development, and also more general cell engineering services. So let's go on to the main focus of the presentation, functional genetic screens. And while there are a number of approaches to be able to disrupt a particular gene or on a gene-by-gene basis or protein-by-protein basis and see what the effect is on the system that you're looking at, there are really only two practical approaches to do this type of analysis on a genome-wide level, and that's using RNAi or CRISPR. Because with both of these technologies, you can go into a database and for all 19,000 protein coding genes or whatever set of genes you're interested in, design reagents that you can be pretty confident are going to disrupt or interrupt or somehow disturb the function of the target of all of the target genes that you're looking at. As a result, then, it's possible to make large libraries that can be introduced into populations of cells and knock out genes across the entire population, different genes in different cells, and then be able to analyze those populations and identify which ones were important for a particular response or phenotype that you're interested in. As a result, then, with CRISPR sgRNA or RNAi short hairpin RNA, we have the ability to make large libraries in lentiviral vectors that can be introduced into a cell population, and then that population can be treated or incubated or sorted in different manners depending on the phenotype that you're interested in. And then the sgRNA or shRNA sequences from those cells can be extracted and analyzed. And depending on what happens to that population, you're able to then identify changes in that distribution of guides or short hairpin RNA, and then understand which genes are important based on the target genes that those guides or hairpin RNA are directed towards. So then, to go into a little bit more detail about how these libraries are made and used, the first thing is to design a set of shRNA or sgRNA targeting the set of genes that's of interest, and this can be genome-wide for 19,000 protein coding genes in the human genome, or it can be a small set of genes of, say, 400 genes that are kinases that are of particular interest. The oligos are then, after design, they're synthesized. We work with Agilent and they synthesize it as a pool, and so all of the oligos are together and then cloned into the library, and then cloned into the lentiviral vector as a pool. And the lentiviral vector, of course, has other elements such as we usually use puromycin selection, but some sort of antibiotic selection, fluorescent protein, etc. And then these lentiviral vectors can be packaged into lentiviral particles. And then you have a library now of lentiviral particles that can be introduced into a population of cells. The reason for using lentiviral particles is because lentiviral infection, or viral infection generally, is very homogeneous. And so a collection of lentiviral particles placed on the population of cells will infect the cells across the population relatively evenly. This is different than transfection, for example, where it's very stochastic and you can get some cells that pick up a lot of different vectors and other cells that really don't pick up any. And so you get a lot of variability from cell to cell. So with a low number of lentiviral particles compared to the number of cells you have on the plate, you can then create a population where you have a single guide in most of the cells have been infected with a single guide across the population. And so then after the cells are infected, you can incubate the cells over a period of time, do various treatments, etc., depending on what the type of screen is that you're running. And then when that's completed, isolate genomic DNA amplify up the guide sequences from all of the cells in the population, from the isolated genomic DNA, and then NGS sequence these, next generation sequencing, to get the counts or the number of each guide in the population across the population. And that gives you sort of the representation of each sgRNA in the whole population, and yet that can be compared then with the original library, or depending on the type of screen, maybe with treatment and untreated populations. But the point is, when you do that comparison, you find that some guides are either overrepresented or underrepresented relative to the original starting population or the control. And in this case, for example, this is a waterfall plot where each one of the lines on the plot represent the change in the fraction of that guide, one particular guide in the population from the baseline or the original library as compared to the population, the experimental population grown for several weeks. And so you can see here that a number of the sgRNA are underrepresented in the experimental population. They've been depleted. And that's because the genes that they target are important for growth of the cells in whatever conditions these cells were grown in. And so they then are essential for growth, and so when they're knocked out, the cells do not grow so well, they die out, and the sgRNA becomes depleted in the population as a whole. This is one of the most common screens that's run with these type of libraries, and it's to look at, say, for example, with a certain treatment, the genes that are required for the cells to survive in that treatment, where when you knock them out, the cells become more sensitive, and that can give you some indication of maybe which genes are important for resistance to a particular drug treatment. Or if you're looking at various genotypes, you can compare cells with one genotype compared to a different genotype, cells with a specific genotype compared to other types of cells, and look for essential genes or essential vulnerabilities in the cells with a particular genotype, say for a particular type of cancer. In addition to the dropout viability screen, there are other types of screens. You can also treat a population of cells and try and kill most of the cells with a drug and then see which ones survive. And that will tell you, for example, which genes are important for that response that you're looking for. So if you're looking at apoptosis and you treat with an apoptosis factor, kill most of the cells, if you have surviving cells, then that may indicate that those genes are important for that pathway to activate apoptosis. And if you disrupt them, then the cells are no longer sensitive to that induction of apoptosis. And then you can also do fact sorting too. So there's you can have a population of cells with a reporter that is either activated or a reporter that's turned off, and then facts sort based on that response of those cells and look at the two populations and compare which targets or which genes are important for the activation or inactivation of a reporter or something similar to that. And so with that, we come to this example of a screen here from a lab at Novartis a couple years ago, where they were looking at non-small lung carcinoma. And about 40% of these cancers have a mutation in the EGFR, epidermal growth factor receptor, which is tyrosine kinase. And so the treatment then for these cancers is erlotinib, which is a tyrosine kinase inhibitor. And so what they wanted to do, though, was understand how resistance to this inhibitor develops over the course of treatment, and maybe what could be done about it. And they were looking actually for both genes that are required for the compound's effect. So, certain genes need to be activated in the cell in order for the compound to work. And on the other hand, other genes may give rise to a certain amount of resistance by working around the compound's mechanism of action and activating other pathways in the cell. And so they wanted to look, in this case, at both sides of the spectrum. And they did this using a cell model, HCC827 cells that were expressing Cas9, and they had that pre-transduced with Cas9. And then they were doing it, they looked genome-wide, so they have a genome-wide library with 5 different sgRNA to each one of the target genes. And then they infected their cells, did pure mycin selection, and they have a the pool, and then allowed that to grow over 21 days. And they split that into the DMSO control versus the 1 micromolar erlotinib treatment. And what they were looking here is in the erlotinib cells, the 1 micromolar treatment, these surviving cells, understanding what sgRNA are overrepresented in these cells that allow them to survive, and also what sgRNA are then missing in this population that when they were knocked out conferred resistance compared to the original or compared to the control DMSO. And these are the results of the screen. What you can see is each one of these circles on the graph on each side is the sgRNA for targeting a particular gene. So each one represents a gene target. And what they did is they sort of rolled up the scores of the sgRNA depletion level on the downside here on the left side of the slide, or sgRNA enrichment level relative to the control on the right side. And then they also have the z-score in terms of the significance of that compared to the rest of the guides plotted in the x-axis. And so what they have then is on the depletion side here are genes that when they're knocked out confer sensitivity then to erlotinib. And so they're missing then in the population that was sensitive to the drug. And so these are ones that are important for the efficacy. And then over here on the right side are ones that are enriched. So when these genes are knocked out, the cells then become more resistant to erlotinib. And so the question is, what are these doing, in the cell population? And so what they found are the hits for sensitization that when they knocked out the targets make the cells sensitive to erlotinib, cluster into 4 main groups. And the GPCR, G protein signaling, when you knock out some genes in that pathway, it makes the cells more susceptible to erlotinib, or the ones that are resistant then lose their resistance. And that's maybe not so surprising. But then also the YAP-mediated pathway and the SAGA transcriptional complex are also other avenues where cells may be able to work around the treatment and compensate for erlotinib. So when you knock out particular targets in these pathways, you can also then regain sensitivity to erlotinib. And then on the other side of the spectrum, they found 3 different pathways that were important for the sensitivity to erlotinib. The PIK3 TOR signaling pathway, again, maybe isn't that surprising, but ubiquitin is something that's a different pathway, and it has to do with protein degradation. And you'll see that one come up again on another study that I show at the end. But this just demonstrates basically the type of analysis that you can do to identify genes that are modulating the effect of a particular drug or treatment on cells. So here's another example of a study that's looking for targets that modulate the effect of a treatment. And in this case, it's for large B-cell lymphoma, the most common type of non-Hodgkin lymphoma. And chemotherapy leads to a cure in half of the cases, but the other half that do relapse have a poor prognosis. And CC122 shows some promising clinical activity. This is the drug that they're looking at. And it seems to stimulate the degradation of IKZF1 and 3. This is Icarus and Aeolus, I think, are the names of the proteins. But that results in T-cell activation, but then leads to apoptosis and growth inhibition for malignant B cells. And so what happens then is they want to try and understand better the mechanism and also try and understand how that's working to have that effect on the cells. So what they did is they ran a genome-wide screen, Crispr screen, on a cell line SUDHL4, which is a cell line that requires either IKZF1 or IKZF3 to be active for it to continue to propagate. Otherwise, it will then apoptose if both of these targets are knocked out. And they have Cas9 expressing cells, transduce the library, again, DMSO treatment versus treatment with the compound that they're interested in. And then after a couple of weeks, they do genomic DNA extraction, amplify the guides, and do next generation sequencing. So the results of the screen are shown here, and what they did in this case was treat with a strong enough dose to kill most of the cells, and they're really interested then in the target genes that are required to be active in the cell for activity of the drug or the apoptotic response of the drug. So in this case, when they knock out a particular target gene, any of these shown here, what happens is the cells then become resistant to the drug because they no longer respond apoptotically when the drug is introduced. And what they found are basically two sets of targets. One set were inhibitors of protein inhibition, which is maybe not too surprising since the drug works by stimulating the degradation of the two targets. And so if you interfere with the ubiquitin pathway that degrades the IKZF1 and IKZF3 targets, then the drug is no longer effective. But the other one was the NF-kappa B signaling pathway that needs to be active in order to stimulate apoptosis in response to the drug too. And so that may then offer an alternative treatment in terms of trying to make sure that cells are receptive, or at least be able to profile tumors to see which ones would be receptive to this particular drug, and maybe which ones would need then some other sort of treatment. So again, this is just basic data from a single screen, but it points to the possibility of ways of then continuing to develop treatment programs for this type of disease in this case. So we just saw a couple of examples of genome-wide Crispr screens, and we offer a couple of libraries, both for human and for mouse, to do these type of genome-wide analysis. However, we also do have an active custom service to make libraries that are more targeted to smaller sets of genes, or that may have variations in terms of specific features in the vector that are required for the screen, or maybe some modifications of the sgRNA depending on the type of screen. Custom libraries are actually a very important part of the services that we provide, because there really are a number of different reasons that it's almost impossible to make off-shelf libraries to cover all different types of screens that people are interested in. And so in some cases, for example, specific sgRNA structures are required, as is shown here on this slide. Are some examples of modified Cas9 binding regions of the sgRNA. In this case, these modifications incorporate sequences that are compatible with capture sequences on 10x single cell analysis platforms, beads. And so if the libraries are modified with these capture sequences, then the sgRNA can be captured in the 10x system, and then it's able to be analyzed as part of the cDNA when you're doing the transcriptome analysis on that system. However, that's just one example. There's other reasons that people may want to try different modifications of sgRNA, maybe to optimize for a particular application or system that they're using. There are a number of different CRISPR variation systems that have different Cas9 nucleases, different Cas-based nucleases such as Cas12, CPRF, etc. And then there are also, of course, reasons to make custom libraries where you want to have vectors with certain specialized features in the vector, and so that's another type of library that we make. And then there's simply the reason that depending on the model that a researcher is looking at, they may not be able to use a genome-wide library. They may need to have a smaller library that is suitable for that model. This often comes up in the case with in vivo systems, and I'll show you that in a moment. And so with regard to using a model system where a full library screen is not really possible, but you want to use a smaller, more targeted screen with a specific objective in mind. Here's an example from Mirati, a biotech company in San Diego, who has a compound, MRTX849, which actually targets RAS. And that's interesting because RAS has somewhat of a notoriety in being a gene that's not targetable because they haven't been able, it's been very difficult to identify treatments, therapeutic compounds that will target that particular gene, or target that particular protein, I should say. And so what they wanted to do was, in testing this drug, make sure that the mechanism of action that they were seeing in the mouse models that they were using, etc., was consistent with their understanding of the mode of action of the drug, and be able to show that, for approvals. And so the problem, though, with an in vivo screen, where you're looking at tumors, engrafted tumors in mouse models, for example, is that when you transduce the library and then inject that into the mouse model, only a small number of those injected cells, or a percentage of those injected cells, actually will grow up to form the bulk of the tumor. And this is a study that we did a few years ago with Amgen, where we're using essentially a barcoded library. This isn't sgRNA that's expressed. This is just a library with about 25,000 barcodes. And we took 10,000 cells and put them in the mouse, and then 10,000 cells on the plate, incubated each, and then looked at the distribution of the barcodes from the cells that were injected versus the cells that were versus the cells grown on the plate. And what we found, for example, is that in the injected cells, about 7% of those original 10,000 cells formed 82% of the tumor. 82% of the cells in the tumor itself were just from a small percentage of about 7%. In another model, 6.5%. And so the challenge is then to, if you're going to run a screen, is to get the entire library that you're looking at in the small percentage of cells that are making up the bulk of the tumor, so that you can actually look at differences in growth. Or if you're looking at, for example, depletion, you understand that the depletion isn't happening because you don't have good enough representation of that particular guide in the library in the tumor, but you have to have enough adequate representation of all of the guides in the library in the tumor. So as a result, really, for these sort of in vivo type screens, you really need to have a smaller library, or a more targeted library. And so that's what Mirati did, is they have a library that's targeting about 400 genes, and these are all involved in KRAS signaling. And so what, again, they want to do is look to see that they're getting an effect on these genes, or changes in these genes that are consistent with the mechanism of action that they understand for the drug. And they did this in a large number of different cell lines, and they did it both in vivo and in vitro in parallel. And so they generated a whole lot of data. It's actually a very interesting paper, and the screen is only one part of it in terms of characterizing this particular compound. But they transduce, for example, H2122 cells is one of the cells that they use. They did also some other cell lines. And then from the screen, they analyzed the results and looked at the changes in the in vivo samples versus the in vitro samples across the couple of cell lines, but this is just the H2122 example, to see which genes were depleted in the tumors and which genes were also enriched. And they found that the genes that were depleted, and the ones that were consistent with the mechanism of action of the compound in disrupting the KRAS pathway, are shown here. And they made the cells more sensitive then to that particular drug. And that makes sense, right? It's KRAS, for example, MYC, and some of the other related targets, mTOR in that pathway. And then a couple of them that are resistant, that when they were knocked out are no longer, the drug is no longer effective, then are NRAS and KEAP1, and these are associated with classic tumor suppressor genes. So this is sort of outside the KRAS pathway that they were looking at. And so then this study then helps confirm that their drug is actually acting and creating the changes in the tumor cells that are expected and consistent with its mechanism of action. At this point now, I'd like to switch gears a little bit and talk more about barcoding. I mentioned in the previous couple of slides that we had done a barcoding experiment a few years ago with Amgen, looking at the distribution of cells that are injected into mouse xenograft as compared to what grows in culture, and how the population then divides and is represented in that tumor versus in cell culture. And the idea with the cell barcoding libraries is that using a very similar approach that we use to make the sgRNA libraries, we can make complex libraries that have particular sequences that are just simply individual unique sequences, barcodes, that can then be used to label populations of cells or label single cells in a population. And then as those cells expand and grow out, you can track the progeny from those labeled cells and look to see if there's biological differences between certain subpopulations in under different conditions. An example of use of one of these barcoded libraries is from MD Anderson, a group at MD Anderson, who a couple years ago created a PDX cell line from a primary tumor going through mouse, and then plated that, and then infected it with a library that had approximately 30 to 40 million different individual barcodes. This is our clone tracker library, our original clone tracker library. And then they did selection and passaged that, and passaged it through many different expansions, continued to passage these cells until they got a stable population. So this is more than 12 passages in culture. And they were able to then stabilize it so that they had a range of several thousand barcodes that were in the population of cells that was pretty consistent as they continued to propagate those. And they were also able then to do a similar type thing by re-injecting and creating xenografts to get clonal, what they call clonal replica tumors, also in different mice. Again, where the distribution of barcodes was pretty stable across each one of the animals or across each one of the cultures. And then what they were able to do is go back and do treatments. Then they have a control where they know the distribution of the barcodes, but then through a couple different treatments of drugs, they can see how that disrupts or perturbs the various subclonal populations in that original population. So some of those populations will continue to grow and expand and maybe have some resistance to a drug. And then when you do a different drug, you may get, if you get a similar set of clones that are resistant, that would indicate the mechanisms are similar, or at least the mechanisms of resistance are somewhat similar. If you get a different set of clones, then that would indicate that there's a different population of cells in that tumor that is confers resistance, that has some resistance to the drug B versus drug A, and they're different. And so they could do this type of analysis, and then we're actually able to break down and see different populations that were able to respond differently or similarly to a couple different drugs, and then went through and did the analysis and actually looked at the expression profile of some of these subpopulations to try and see what are the differences and what might be leading to this resistance. So that particular study, which I encourage you to go look at, it's pretty astounding what they were able to do. Used our original library, our original barcode library, clone tracker, where the barcode is introduced into the cells and then integrated into the DNA. And so in that case, then you look at isolate the DNA and amplify the barcodes from the DNA in a cell population, and you can look at the distribution of barcodes in those cells. But that library does not have is that the barcode is expressed in the RNA. And for some studies, especially if you're looking at expression analysis now with single cell systems such as 10x, it can be very useful to also have the barcode expressed in the RNA so that it can be captured and read as part of the cDNA while you're looking at also the expression profile of the transcripts in the cells. And so we have sort of our next generation or recent generation of clone tracker libraries where the barcode is expressed. So these are clone tracker XP libraries for expression. And the barcode is expressed in the 3' untranslated region downstream of the drug resistance. So when you introduce the library into cells, you do it at a low MOI, so you only get one barcode per cell. It integrates into the genomic DNA, and you have the genomic DNA labeled for those cells. But also then it's expressed on the transcript with a puromycin resistance, and then can be detected when you are using a 10x system or you make cDNA with any other type of system. And so you can detect it in both the RNA and cDNA. The RNA, cDNA, or the genomic DNA. And so an example of this expressed barcode idea is shown here with a small pilot study done by a group at Genentech. And this was just a preparation of a larger, more involved study with a complex barcode library that I'll show you in a moment. But what they did here is they labeled 5 different cell populations. So these are just standard cell lines, PC9, MDA, MB231, MCF7, sorry, not MCF7, MCF10A, but they're just different cell lines. And what they did is they had a culture of each, and they introduced barcodes into each one of these 5 different cell lines. And then they mixed them all together. And then they ran the 10x system and profiled the single cell RNA expression for each one of the cells in the system. But now they're all mixed together, so there's 5 different cell types in this analysis. And then when they did the expression profile, they were able to get 5 different cell types clearly defined in the expression profiling. And this is this is exactly what's expected, because each one of these cell types has a different expression profile, and it's well known what they are, and they can go through and they can separate them. And so then they also went through and looked at the barcodes in each one of the cells on the 10x system. And what they were able to do then is using the barcodes, the cell barcodes that they introduced initially, and they were able to then compare them to the expression profile that they got from the 10x system based on the bead barcode in the 10x system. And they were able to see that they correlated almost 100%. And so they got very good resolution between the two. So they were able to use the barcoded cells. And the point is here, again, it's a simple experiment, but they can then take the expression profile that they're getting from the 10x system and then correlate that back to particular populations of barcoded cells in the original population. And so the idea then is to do this in a much more sophisticated way with a more complex library, where you have a population of cells that are transduced with barcodes. And so you have a way of identifying what are the various sub-clonal populations within this population. And then you take that population and you treat it with a number of different treatments. And then after treatments, you can see what the different responses are in the population and identify if there are a certain set of barcodes that respond one way to a particular treatment, and then another set of barcodes that respond a different way to a second set of treatment. And so then using those barcodes, then you can go back to the original untreated population and take a look at what is special about the cells that responded in one way to treatment 1 that say didn't respond to treatment 2, or vice versa, the ones that in that original population that responded in treatment 2, and see what pre-existing features, and especially in this case using a 10x system, to see what pathways are maybe active or not active in certain sets of subclonal populations in that original population that allowed them to, that predispose them to be able to manage treatment 1 or be able to respond a certain way to treatment 2. So what they did to do this screen is they created a library that's very similar to the Clone Tracker XP library I showed you. It's slightly different design, but basically same idea. At the 3' end of the selection marker, in this case Puro EGFP, they have a barcode that is expressed then on that transcript and is detectable in the RNA. And they use this to transduce a population of cells, PC9 cells, which were then treated with 3 different compounds. Erlotinib, which is the EGFR inhibitor that we talked about previously. And so in this case, they also then have an analog which they wanted to compare with erlotinib, which stimulates the degradation of EGFR also, as well as just binding it and inhibiting it. And then they have a control version of that, which is the GNE069, which does not enable degradation of the EGFR. It actually prevents it as well as inhibiting the tyrosine kinase activity. And then they have an untreated arm also in the screen. And the idea is to be able to see if they can distinguish between the drug-sensitive versus drug-resistant clones for each one of these variations in the population, and see if there's a difference in the clones, especially with respect to the erlotinib and then the one that also stimulates degradation, which is the analog of erlotinib also. So what they did then is they transduced the library into their cell population, and then took about 600 cells, and then they expanded them through several different doublings, at least 12 different doublings, and then took the untreated population and analyzed that with the 10x system, and then did 4-day treatment on the 3 different variations with the 3 different compounds, and then also ran those through the 10x system. So when they looked at the results, they found that the baseline population had about 551 barcodes, which is pretty consistent with the 600 cells that they had started with, and that each of the treatment, the erlotinib and then the 2 GNE analogs, resulted in a lower number of barcodes. They decreased the diversity of the population. And so then the question is, what's different, especially between the degrader, the GNRE104, and the inhibitor, the 069 variation? And why is the 069 more effective than the 104 in terms of killing more cells or more populations of the subclone, right? Getting less barcodes. And so they then looked back at the original baseline population and looked at the barcodes that survived the treatment, or the ones that died off in the treatment, depending on both of the populations they can look at in the baseline. For the GNRE069 and the GNRE104 populations. And what they found is that the most significant difference in gene expression had to do with the endoplasmic reticulum processing of degraded protein, so protein processing in the ER. And they were the pathways that were most significantly had differences in expression levels between the 2 populations, the degrader cell treatment cells and the ones that were the resistant the analog that was resistant to degradation. And so what they found then is that basically this being the most significant between the 2, and that blocking the degradation of the EGRF receptor was what enabled the GNRE to be more effective. And so what they surmised from this is that basically the EGRF inhibitor response requires induction of ER stress also in order to be maximally effective in terms of the treatment. And this is just one more example that I'll quickly show you that combines barcodes with sgRNA. So the previous two examples I showed you are just really barcode experiments with barcoded libraries. In this case, though, it's a small sgRNA library with guides that are targeting tumor suppressor factors. And in addition, with the way the library is constructed, in addition to have the guide being expressed, it's expressed in conjunction, the library is cloned in such a way that in conjunction with the guide, there are a series of a few 100,000 barcodes associated with each guide, so that each one of these guides has different sets of barcodes, or different barcodes, but the point being that each sgRNA and barcode combination is unique for each one of these tumor suppressor guides in the library. So what does that mean? That means that for each infection of the construct, you have an sgRNA going into a cell. It goes in with a different barcode, and so you can keep track of then each individual infection when you're looking at the population at the end by the number of different barcodes you have associated with a particular guide. And in this case, it's an in vivo experiment. This is by a group that was at Stanford, and they've since gone on to form a company called D2G that we work with, on a number of different constructs and libraries. And what they did then is they took this small library. I say small library, but it's actually a large library. It's just a small number of tumor suppressor guides. As I said, there's 15 of them, 15 targets. And they have the mouse inhale that, and so then the library, this is a lentiviral library, enters the lungs and it infects the cells around the lungs. And since each one of these is individually barcoded, they can take a look at, for example, at P53, the guide targeting P53, and they can actually see by the number of barcodes how many cells it entered in terms of the tissue of the mouse by the number of different distinct barcodes associated with that same sgRNA to P53, and they can do the same for each one of these guides to the different tumor suppressors. And not only that, in terms of the absolute number of barcodes that are associated with a particular guide, give them an indication of the number of cells that were infected, but then the representation of a particular barcode sgRNA pair, if that's represented in large number, that indicates that that particular infection grew out to an extended amount, and so the size of the tumor then can be estimated by the number of barcodes that are being read in the NGS. And so in this experiment, for example, you have P53, and you can see that you get a number of large tumors with P53, and can compare that with, say, the overall infection amount, and also with LKB1, and also with the various other targets that are knocked out, these tumor suppressor genes that are knocked out and then lead to tumors. And so what you have inside one animal is basically you're modeling several different tumor genotypes. And then, of course, the next logical step is to do treatments of those animals with different compounds, and you can see that certain sets of drugs will have an inhibitory effect, and other sets of drugs they're resistant to, depending on the genotype. And so you can test different treatments and different combinations of treatments in an animal in multiple phenotypes with just simply this one library, using a combination of barcodes and sgRNA. And so with that, I'd like to say thank you for your attention. And again, I'll turn it back over to Ryan, and I'd be happy to take any questions that you would have or any comments in the time that we have left.